and obtained MA and PhD from Cornell University, New York, United States, and completed postdoctoral fellowship from the Harvard Medical School, Department in Molecular Neurobiology. And then she is now working in the Chai University Medical School, Department Biotechnology department. She is working now, and today her topic is about skin recall, passing exosome connecting skin recall reprogramming, and then we shall give a wonderful lecture. Thank you.
Thank you for your introduction. My name is 제45차, and thanks a lot for having me here. This is first time I have this kind of conference, so my talk might be a little bit different from previous one. I'll maybe talk about exosomes and stem cells and how they recode our old skin cells. A little bit scientific. I'm not a doctor. I'm a PhD. As introduced by the organizer, I graduated Cornell Neuroscience Statistics. I moved to the Boston Medical School, Harvard Medical School. I studied stem cells over there. I moved back to Korea 2009. Since 2009, I'm in professor at Chai University. And I work government side. I'm a committee member in scientific field in government side. Okay, as I said, 2009, I came from USA. I developed mostly stem cells, like fetal neural stem cells, and for the Parkinson disease, mesenchymal stem cell for the Alzheimer's disease. Those are now on clinical trials, and using stem cell therapy, I've learned a lot. And I feel like the stem cell development is very hard to be outside, like to the customers. It's expensive, but the process itself is very, very hard. And about 2015, I realized that exosomes from stem cells are really, really similar as stem cells. And stem cell, the previous speaker gave a lot of good introduction about exosomes. And from that 2015, I extended my research with exosomes, and today I'm going to talk about mostly exosomes. You might learn what are the exosomes in skin booster. And 2014, this year is very important. February last month, I established my own company called Record, and I developed cosmetics using exosomes. I will introduce to you today. Exosomes, we have to talk about stem cells first when we talk about efficacy of exosomes, because exosomes have stem cell contents. So let's think, many people thought stem cells are the one. Stem cell can rejuvenate our old skin cells, old cells. But think about stem cells, how can they live in the cosmetic environment? Stem cells alive, once they put into the cosmetic conditions, they will die. So when you call stem cell cosmetics, all liar. They're not that stem cells alive in the cosmetic situation. But exosomes are very stable, and it can be game changer. That's the reason I focus on stem cells in skincare. So when we compare between exosomes and stem cells, exosomes have similar characteristics of stem cells and very stable. It's very stable. As previous speaker said, depends on the storage condition. It can last like 2 years on the room temperature. That's very fancy. And it's stable, easier, and also have efficacy. And then why not? We should use exosome for the cosmetics. But stem cells, very efficacy, but it's not really stable. It's live stem cell, very hard to regulatory point of view. So exosomes are better, I can say. So what are the exosomes then? When exosomes first found, people thought those are trash. It's just nothing. And it's like 30 years ago. But 10 years ago, about 10 years ago, people study with microscope, nano size of exosome we cannot see with naked eyes. With a microscope, they start to find, oh, exosomes are real. It's bilayer, like a membrane, cell membrane is bilayer. They have all the contents like a microRNA, small RNA, protein growth factors like a stem cell have. Even though it's nano size, these like a vector of cells. And also surface markers, they're very fancy. So what they do usually in our body system, they are communicator. Actually cells cannot speak, but exosome is like a cell's mouth. They communicate between cells. I'll explain a little bit. And also, as I said, they have genetic materials, meaning that this genetic material can recode or reprogramming your old skin cell to the young skin cell. That's the mechanism. So, as I said, donor cell have exosome in it. Usually, all the endosome, multibody, multivesicle body, let endosome. Exosome come from these endosomes. Researchers call exosome secreted by all the endosome, late endosome. They call exosome. So exosome come from the donor cell, and that cell move to the next neighbor cells. And it's very important how the exosome can enter the cells. Cells usually don't like anything from outside because they can be really harmful. But exosome, receptor-ligand interaction, endocytosis, membrane fusion, with these various mechanisms, cells can come to my home. They are really pleasant to get exosome into their cells. Meaning that when you treat like a microRNA or messenger RNA, DNA, once your body have that natural material, your immune system will immediately kill the messenger RNA. But once that material put into the vesicle, your body thinks those are really natural. Meaning that whenever you want to give stimulator, like a messenger RNA, some protein, you have to put into the vesicle. That way, your cell will love to enter their cells. That's very important for me. Okay, so exosome, as I said, is a vector of cells, meaning that all the materials are same as the original cell. So let's say if stem cell have their exosome, the stem cell derived exosome have same efficacy of stem cell because the materials are same. Because of that, exosomes have use for the medicine, like a vaccine, cancer therapy, gene therapy altogether. Also, the application getting expanding, like diagnosis, neurodegenerative disease, and as aging, immunotherapy, etc. Very getting expanding. So actually, I'm a general secretary of kccs, Korea Society for Exosome. Also, I'm an IOS member of ISAP. That conference is getting really expanding, and even their journal is a very high impact journal, meaning that this field is getting really explosively expanding. So challenge of exosome utilization, it's a little bit hard to mass production. It's the same as stem cell. Stem cell is very hard to expand. That's the reason stem cells are very expensive. And even exosomes are a little bit hard to make a mass production. And still, we need to develop more strong efficacy in terms of anti-aging potential. And exosome can make a targeted delivery by engineering the membrane. It's a little bit hard. Okay, then what are the exosomes' role in skin rejuvenation? Let's say, this looks like my skin. It's getting wrinkly in it. used because of this bad radiation, and because of that, inside of a cell have pro-inflammatory factors and a lot of bad things around. But as I explained, exosomes have the fixing material or stimulator to the endogenous stem cell. Once you put the stem cell into the skin, that will correct those problems, because those stem cell or those microRNA degraded immediately those pro-inflammatory factors, which I have data today also. So that is a basic concept. So conceptualization, as I mentioned, and formulation is very important to make a prolonged, stable exosome. And to make a realization, we talk about the market really precisely or scientifically and easily. Okay, so impact of exosome technology and skincare. We try to make enhanced efficacy in targeting, also skin communication. Critical evaluation of skin recode is all based on regulatory compliance. Since the exosome is kind of bioactive material, we have to make or produce this exosome from GMP facility, not from lab or something. Okay, so I would like to introduce recorded skin science. We've developed placenta-derived exosome and various kinds of stem cell exosome, also plant exosome, because plant exosome we can immediately use without FDA approval. So we start with the first exosome first. So what is the main component of exosomes? We sequencing proteomics, RNA sequencing, and see what kind of materials are there. And then materials are similar as the animal cargoes. Okay, so record on highly observant exosome complex. This have nucleic acid we found, and also I would like to say that from this part, those results are not published yet, so please don't take a picture a lot. You can take a picture, but not really record like a video clip. Okay, so our exosomes we characterize have, we isolate UC ultracentrifugation or TFF system. Their size are morphology are exosomes, and their size are really exosome size. Also, the tip material is like same as the previous animal exosomes. We compare with animal exosomes; they're really same as animal exosomes. Also, we found that we analyzed proteomics. Even for exosome have human proteins when you compare with matching. So they have those proteins are NICEP guideline 2003. So this G protein, Hisha protein, those are proteins NICEP said, oh, if exosome have that protein, they are exosomes. So we can call this fruit exosome really exosomes. Also, we analyze their function. We put the H2O2 is really stress condition. When we put the H2O2, so H2O2 here, and as you can see, very bad oxidants goes up. But once we put the exosome in it, those are normalized, like I said, very strongly normalized. Also, we check the protective effect. Protective effect is like that. Let's say, I'm not that old enough, even though I'm old. I want to protect my skin before my wrinkle really deep inside. That's protection effect. So in this condition, we put the first exosome first and then add H2O2 later. So hopefully our hypothesis is that first exosome will prevent these bad things and prevent our wrinkle. So as you can see, really strong protective effect found. Also SOD1, SOD2 are really strongly reduced. And also when we check the anti-inflammatory effect, they are really Anti-inflammatory effect and antioxidant effect, they are really strong with Exosomes. Also, we check the anti-aging effect. Let's say already I have wrinkle in it. It's not protection. We have to get rid of this wrinkle. And in that case, we put the HO2 first and then add for Exosomes. As you can see, senescence markers reduced, meaning the cells are still young, alive. And also we check their microRNA in that for Exosome. We do RNA sequencing, and mostly 159 microRNA, 56% included. This 159A microRNA are really important for the PDGF signal pathway and Wnt pathway, meaning that they will regulate the bad genes in skin in all the situations. And then we will also do clinical trial and compare the previous one. 2 weeks, only 2 weeks after, we can find their wrinkle get improved and their tone improved too. Significantly increased, like less than 0.05, and this is quantification of that effect. There is no side effect. So, based on those data, we make a record cosmetics like a record anti-aging. We have toner and serum and cream all together. And if you need or learn about, if you need some Exosomes like fruit or stem cell, let me know. I have a lot of different kinds of Exosomes in my lab and company. So this is a summarize of the Exosome: energizing, regeneration boost, and moisturizing effect, skin tone improvement, wrinkle, fine line reduction, skin defense enhancement, calming, brightening, skin barrier strengthening, and antioxidant actions. This is acknowledgement I'm working on. I actually I'm an expert on neuroscience point of view, not skin expert. But since I'm working on the Exosome, I first developed skin, and I'm developing hair and liver. You know, very interesting phenomena is that when we put the Exosome in it, the Exosome go into the liver, and turns out liver getting really better. So because of their finding, side finding, developing Exosome might be good for the liver. You know, everyday, not everyday Korean people like drink alcohol, so we need some prevention effect for our liver condition. Thank you so much.